Right. Komoda Lamai, are you okay? Good evening, good evening. Komoda eating. Right. So what's news? No school these days, ne? No school, ne? On holiday. Right. Can hear, ne? Good evening, good evening. Right. People, you got paper number 7, ne? Paper number 7. You had chemistry Eve. You had chemistry Eve today? Yes or no? You have it, ne? Yes. What time? 6 to 7. 6 to 7. 6 to 7:30. What's the time? 6 to 7:30. Yeah, okay. Right. So half an hour break, ne? Half an hour break for bio, ne? Right, people, paper number 7. 6:30, 7:30. 1 hour only. That's easy, ne? So I'm the one who's taking a little longer duration, ne? Right. All right. So people, paper number 7 I have given you, but it's we are not going to discuss the paper today. We want you to submit the paper. We want you to submit the paper, and we'll give the marking scheme on Saturday. Discussion will be on next Thursday. This paper, right? Today is just a preparation. What do you think is like difficult, people, for the in the paper? Did you go through the paper? Did you go through the paper? Paper is mainly regarding the resolution power of the microscope. Cell structure function, neither. We learn, people, cell cycle lesson. Cell cycle lesson we discuss. Cell structure function. I don't have any plan to discuss cell structure function. Thought of like discussing with you fungal diversity, but that we discussed very recently. Neither. Fungal diversity we learned very recently, but anyway, thought this can be little interesting and a little confusing part for you all. So I think it's always worth of discussing fungi couple of times, right? Because you forget them easily, ne? Fungal life cycle. So our today's aim is to spend some time, people, discussing fungal fungi, ne? Fun guys. Now, people, fungi. Right, fungi, 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 ne? Fungi, fungi. You can say fungi or fungi. Fungi. They are prokaryotic or eukaryotic? Are they prokaryotic or eukaryotic, people? Fungi. Eukaryotic with collagen. Eukaryotic with collagen in extracellular matrix. Who's that? Who is that? Collagen? No collagen, ne? You can't say collagen. Who's having collagen in the extracellular matrix? But anyway, these guys are having cell wall, ne? We don't call that as an extracellular matrix, but some extracellular content, extracellular materials, right? But collagen is not a present. Collagen is present in animal cells, among animals, right? Danishas is mammalia. No, not mammalia. We said animalia. Animalia. Animalia, not mammalia. Mad. This is like now diversity kottu ne. You now make diversity kottu takatatakan, 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 right? And you mix it up, right? Takatatakan, takatatakan. Diversity kottu. And then people, what is tasty, ne? When you make kottu, tasty, ne? Right? So, fungi, eukaryotic. Eukaryotic. We learn, people, fungal filaments, chitin containing cell walls. Okay, shall we make it into discussion? Can you type me general characteristics of fungi? Tell me, people, the general characters. Chitin containing cell wall is there. What is their nutrition? What is their nutrition, students? What is their nutrition? What is the nutrition? Heterotrophic, absorptive nutrition. Very good. What is called as absorptive nutrition? Heterotrophic, true. All of them are heterotrophic. Yeah, correct. But what is heterotrophic absorptive nutrition? What is the speciality about the heterotrophic absorptive nutrition? They secrete enzymes out. They secrete enzymes out, digest the extracellular materials, and then absorbs it back. So that we call as the absorptive nutrition. Heterotrophic, generally people, all of them are heterotrophic, but you learn people, there can be commensalism, parasitism, commensalism, parasitism, sometimes mutualism, they are also present, right? Heterotrophic, but heterotrophic fungi, heterotrophic, but then we can see people, we can see that decomposers are there. Decomposers are there. What do you call decomposers? They grow on dead materials. Now, I drew a cartoon. This is a dead plant. This is a dead plant. Fungi grow happily. Hugging this dead tree. Then that is decomposer. Decomposer break down complex materials into simple materials. So that is one of the important role in nature. Like photosynthesis is important, decomposition is important too, right? Decomposers, it's important. And then people, we said that some are going to be parasitic, very good. Now, when we say parasitic means they are growing on a plant, hugging the plant, they grow, hugging the plant. The fungus is happy, plant is unhappy, right? Then what do you call that, people? That is parasitism. And then people, we learn commensalism. What is commensalism? Commensalism means the tree is really upset about this, right? But in this case, people, tree don't have anything, right? Neutral. Why commensalism? There is no harm to the tree. Fungi is get benefited from the association, right? And then people, what is the last one? Mutualism. Mutualism, what we say, people, both are happy, like you and your friend. Mutualism, both are happy and both love each other's companion. So that we call as the mutualism, right, people? So we talked about different types of nutrition present among fungi, right? So what is saprotrophic, Kilahano? So what is saprotrophic? Now we learn saprotrophic. Now decomposer is not actually a nutrition. Decomposer is a process, breakdown of complex materials into simple materials. But if you think it as a nutrition method, saprotrophic is the actual nutrition type. So I wrote it as a decomposer, but it is actually saprotrophism. They carry out decomposition, breakdown of complex materials, and then releasing into simple nutrients. That is a decomposer. But some organisms survive by the nutrition obtained by the decomposition process. Then it's a saprotrophic or saprophyte. Saprotrophic, saprophyte, saprotrophic, right? Selvanathan is asking what is saprotrophic. Is that clear? You understood that? So actually, that's like in our syllabus, they have like mess it up. So it's actually has to be decomposer is not a nutritional category. It's breakdown of complex materials into simple materials is the decomposition. But some survives by using that as a nutrition method. Then it is saprotrophism. Right, people, fungi. So that's about fungi. General characters, we learn, people, fungi, they have mycelia. Mycelium is a net-like structure. Mycelium is a net-like structure. Fungal mycelium is a net-like structure. Fungal mycelium is a net-like structure, right? We can say fungal mycelia. Now, you learn terms, people, the hypa. This is hypa. This is hypa. Fungal filament, one filament is hypa. Then many filaments together is the mycelium. Mycelium is network of filaments. The mycelium. Hypa is one. Hypa. Hypa. If you want, you can say hypae. Phae, hypae. Then it's a plural, many hypa. Right? Many hypa is hypae. Right? Hypae, hypa, happa. Happu. Happache. Right? Hypa. Happu. And then we talked about fungal hostoria. Hostoria. What are hostoria, people? Sometimes some fungi make some penetrating filaments into the plant cells, especially people when they survive as parasitic. When they are parasitic, they take the nutrition from the host, and that is the hostoria they use for that. Hostoria, right, absorb nutrients. And then we talk about cynocytic. You learn fungi, they are cynocytic. What do you call cynocytic? Cynocytic means what? What is meant by cynocytic? Yes, cynocytic means what? Septe, septe absent is cynocytic, septe present. So fungi, people, they have cross walls. They have cross walls. We call them as septe. Fungi have cross walls. We call them as septe. This is fungal filament. And these are the nuclei. But there aren't any partition. There aren't any partition in the filament. So if no partition, then people, it is cynocytic. But sometimes we can see this sort of partition available. When a partition or the cross wall is there, people, then we say that it's septate one. But the special feature is, people, large pores are present at the middle. So even nuclei and the cell organelles can flow from one cell to another. Even nuclei and cell organelles can flow from one cell to another. So septae with large pores are available. That makes a special feature in fungi. So, people, this ability allows the fungi to transport nutrients from a vast area. They can collect nutrients and then can concentrate them at one point. So therefore, fungi can gather materials more effectively. And as a result, people, fungi can grow faster. They grow faster. So within a couple of hours, people, sometimes they can grow a fruiting body, right? Within hours time duration. And that's because of this force present on the cross walls. Sometimes not because of the septate. Whether with or without the septate, the fungal mycelium is vast, helping in gathering nutrients from large area. All right, people, we talked about it. That's nice. I think we talked about the basic features, basic. My remote isn't working. So what is hostoria, Kilahano? What is hostoria? Sorry. I think you have to do it manually. Right. So, right. Now, people, we were discussing this fungi, general characters of fungi. So we learn all these features now. We revise them. Eukaryotic cell walls are made up of chitin, absorbed to heterotroph, different species decomposes. Right? We talked about it. Unicellular fungi present. Unicellular fungi present, multicellular fungi present. Right? multicellular fungi. Multicellular fungi. What do you call multicellular fungi? We use a special word for multicellular fungi. What do you call multicellular fungi? We use a word. What do you call? Molds. Very good. DJ. DJ. JD. I saw it as DJ Nisha. No, it's JD Nisha. Right. So molds. Molds are there. And then multicellular fungi produce mycelium. We talked about it, right? Then characteristic features of phylum Chytridiomycota. Chytridiomycota characteristic features. Now, people, we learn chytridiomycotes. In Chytridiomycota, we learn people chytridiomycotes are having life cycle with gametes. They have asexual reproduction, sexual reproduction present. Asexual reproduction is involving in forming zoospores. Asexual reproduction, they produce zoospores. So when you take a filament, fungal filament like this, right? This fungal filament is aseptate. Now this the tip of the filament can form sporangia. The sporangia inside, we can see that the spores are formed. The spore is a motile. We call this is mitosis. Spore formed by mitosis. Mitosis spores are 2N. 2N, what do you call this? Zoo spore. Zoo spore is produced, which is 2N. And then people, this zoo spore grow back to the mycelium. And then people, we learn during the if meiosis occur, the sporangium is forming haploid zoospores, haploid zoospore, the haploid one, haploid zoospore can grow into a new mycelium. This mycelium is forming gametangia. These are gametangia. Gametangia are produced. The gametangia forms male gamete. Gametangia produce female gamete. Male-female gamete unite and then form the zygote. Zygote grow back and then form people the mycelium. In this case, you can see people, this one is the gametophytic mycelium. Gametophytic mycelium. This one is called as the sporophytic mycelium. Now, you don't have to know about these things in detail, but anyway, what you need to know, there are gamete formation in between. There are gamete formation in between. That is actually the main feature, people. Right? Aquatic or terrestrial, some are decomposers while others are parasitic. Multicellular or unicellular, when multicellular cynocytic, produce zoospores which are flagellate for reproduction. Cell walls are made up of chitin. Some of them form colonies with hyphae, while others exist as single spherical cells. And then people, characteristic features of phylum Zygomycota. Characteristic features of Zygomycota. Now, zygomycotes, we learn people, they are multicellular, terrestrial form of fungi. And then people, the nutrition can be parasitic, commensal type of nutrition can be there. And then this Zygomycota is cynocytic like Chytridiomycota. Zygomycota is cynocytic too. That is cynocytic too, zygomycotes. Cynocytic too. Right? And then you learn people, during the asexual reproduction process. Now, if this is the mycelium, during asexual reproduction process, some vertical filaments are going to form. Vertical filaments are going to form people sporangia. Sporangia produces spores. And you learn people, these spores are going to form inside the sporangium. And that is asexual spores. Asexual spores are produced inside the sporangium. What do you call them? Sporangiospores. Sporangiospores. Okay. Sporangiospores. Okay, fine, correct. Or else endospores. Endospores. So the spore disperse and then germinate people and then form in the mycelium back again. And then people, we learn people, if those two genetically dissimilar mycelia are nearby, they are ready to carry out Sexual reproduction. And then people in sexual reproduction, they are forming gametangia. Gametangia has septae. These gametangia are not sexually differentiated. That means you cannot identify males and females gametangia. Gametangia are going to fuse and then form people the zygospore, the mighty zygospore. The zygospore receives these nuclei. So the nuclei are present inside the zygospore. Zygospore is sturdy structure, can pass unfavorable condition. And then inside the zygospore, nuclei are going to unite. And then people, very temporary stage is there where the nuclei are going to fuse with one another. And then have a short 2n stage. The short 2n stage is going to end up soon. And then we can see that people, the zygospore is going to germinate. Zygospore is going to germinate, forming a new sporangium. And these sporangia are having sexual spores. Sexual spores are going to disperse and then forms the new mycelia. Sexual spores are produced by a process called as meiosis. Meiosis occur here. So we can see people, the nuclear fusion happened here. What do you call the nuclear fusion? What do you call the nuclear fusion? What is the term used for nuclear fusion? That is correct, Santosh. Karyogamy. Karyogamy. And what do you call as the union of the cytoplasm? Union of the cytoplasm is called as plasmogamy. And here the plasmogamy occur. Plasmogamy take place. Plasmogamy occur. And then people, meiosis occur. Life cycle is over. Life cycle is over. Zygomycota. Zygomycota, we can see most of them are saprotrophs and some of them are parasites or common cells. Mycelium is xenocytic and aseptate. Septate found only where reproductive cells are formed. Asexual reproduction produce sporangia in which genetically identical haploid spores are endogenously produced, called as sporangiospores. And sexual reproduction, a zygosporangium is produced, which is sturdy structure produced in plasmogamy and karyogamy, and the zygosporangium is resistant to unfavorable environmental conditions. Right? Resistant to unfavorable environmental conditions. Right? That neither. I'm just speeding up like a fast train here. And then people's zygosporangium is multinucleated structure which is resistant for drying and freezing, and they are metabolically inactive in adverse environmental conditions. Zygosporangium produce genetically diverse haploid spores when environmental conditions are favorable. Characteristic features are phylum Ascomycota. Ascomycota. Characteristic features of Ascomycota. Give me one example. 2 examples. 3 examples of Ascomycotes. Zygomycota. Hey, you Zygomycota. Examples. Zygomycota examples. Mucor and what is that? Rhizopus. Mucor and Rhizopus. Right, people? Mucor and Rhizopus. Mucor and Rhizopus. Right, fine. Mucor and Rhizopus. Right, coming back to talk about Ascomycota. Now, these Ascomycotes, Ascomycota, they are septate. Sarara, special feature, partition wall present, septate. From these, vertical filaments are going to form. Vertical filament is going to form. The vertical filament is forming spores endogenously or exogenously.
They form tube-like structures like this and then begin to generate the spores towards the outside like that. So what do you call those spores, people? You call them as exogenous spores. And they are ascospores. Ascospores are exogenous. Ascospores, are they sexual or asexual? Are they sexual or asexual spores, my dear? Are they sexual or asexual? Asexual spores. And there is a special name for ascospores. Sorry, I made a mistake. These are not ascospores. What do you call people them? What do you call them? You call them as conidia. Ascomycota, asexual spores are conidia. What do you call the structure bearing the conidia? Conidiophore, Santosh, correct? Conidiophore. Now, when come to the secret sexual reproduction, they are forming gametangia. This gametangium has a small thread like that. What do you call this gametangium? Male or female? This one. What do you call it? Male or female? And then there is another one which forms a structure like that. And what do you call this? What do you call this? This is male or female? Male or female, this guy? Male or female? That is antheridium. This guy, what do you call male or female? Female. Female you call ascogonium. This is antheridium. Ascogonium and the antheridium makes a touch. That's a magical touch. Like, it's a magical touch. And this magical touch is now transferring. This magical touch transfers. It transfers the nuclei into the ascogonium. Ascogonium stretches and then transforms into ascogenous filament. It transforms into ascogenous filament. This is ascogenous filament, the ugly ascogenous stretched filament. Now the filament is surrounded by other fungi, right? And then this is the beginning of a special stage. This is the beginning of a special stage where the two nuclei now have a ballet dance. Two nuclei are now in a ballet dance. What do you call this mycelium? What do you call this stage? What do you call this stage? What do you call that? Dikaryotic stage. That is good. Dikaryotic stage. Two nuclei are now in a dance. Right? Two nuclei like that. And then people, this filament, new filament, branch and then transform into sporangia, especially at the tip. Nuclei are going to unite. And then people, new nuclei forms the two-end zygotic nucleus is going to form, which undergoes meiosis and forms people spores. How many spores are going to form? 8 spores. What do you call those spores? Ascospores. So in Ascomycota, what do you call this entire structure? What do you call this whole structure? Ascocarp. What do you call this structure? What do you call that? What do you call ascus? This is ascus. And then this nuclei form 4 nuclei and then form 8 nuclei, and you call them as ascospores. This is ascocarp. The whole structure. And then inside you can see many ascuses.
Many ascus present. What do you call the plural word? Huh? How do you say ascus? Asai. So many asai present. We can show another asai here. Another asai here. 1, 2, 3, 4, 5, 6, 7, 8. So many asai are there. So many asai present. And then people, these ascospores are going to disperse. And then people, they are going to begin the new life cycle. New filament is going to form with that. And this is the story. This is the story. You can see people marine, freshwater, or terrestrial, parasitic or symbiotic. Most of them are decomposers. Unicellular or filamentous, multicellular, asexual reproduction by conidia at the tip of the conidiophore, which are specialized hyphae. In sexual reproduction, fusion of the sexually differentiated gametangia take place and produce sac-like structures called asci. Ascospores are produced within the asci. Generally, there are 8 ascospores produced in each ascus. And then most of the fungi produce ascocarp enclosing asci. Right, we come to the last part, only this content. And then we are now people talking about Basidiomycota. We are now talking about Basidiomycota. Basidiomycota. Examples. Who's going to tell me examples of Basidiomycota? Some sort of meatballs are there. Eh? We said meatballs. Meatballs or what? What balls are they? Puff balls, not meatballs. Puff balls. Agaricus. Shelf fungi. Shelf fungi. Right? Shelf fungi. Oh, that's nice. Shelf fungi. So they are parts. They are some types. Shelf fungi. Shelf fungi. So people, if you take asexual reproductive structures are not common, can present but not common, very common. Asexual reproduction structures are not common. And then sexual reproduction occur. Sexual reproduction occur, but there is no gametangia formation. No gametangia. They don't want any sex organs, right? They carry out sexual reproduction by touching the filaments. We call these filaments as the what do you call primary mycelium. When you can see genetically different two primary mycelia meet each other, when they meet each other, they begin their love cycle. They begin their secret love life. And then people, what happened here? The nuclei are going to unite. And then people, we call this as the plasmogamy. That is correct. Plasmogamy occur. And then this is now leading into formation of a new filament, which we call as the dikaryotic filament. Now you can see that here the new dikaryotic filament begins here. There is a special way of forming this dikaryotic filament, which is out of syllabus. We don't talk about how it exactly happens, but it happens. This dikaryotic filament is the dominant stage. It's the dominant stage. And then you can see people, so many vast filamental network is going to grow underground, forming the mushroom. Finally forming this beautiful mushroom with gills on the underside. With gills on the underside, we can see this beautiful mushroom is going to form. There is a cap present here, cap, and the underside has gills. Cap and the gills are there. And if you take one gill, this is one gill, and then some structures are going to form here. Basidia. The basidium has those two nuclei inside. Soon these nuclei are going to unite. They're going to unite. Union of this nuclei is now forming zygote. This is the 2N zygote. 2N zygote undergo meiosis and then form people 4 nuclei. 2N zygote undergo meiosis and then form 4 nuclei. 2 are the black types, 2 are the red types,
two black and two red types. And actually, people, this one further grows and then exogenously form four spores. So therefore, we call these sexual spores as exogenous, or else you can say, people, as the exospores. These are exospores. Exospores are produced towards outside. By growth of these exospores, the new filaments are going to form once again. And you know, this is going to form this filament, this is going to form that filament. The life cycle starts over again. That is the simple reproduction of basidiomycota. Basidiomycota, right? Simple reproduction of basidiomycota. Fit neda? Understood? Right? Moving people to the discussion. Moving to the discussion. They are terrestrial, means people, they are land. They grow on land. They are major decomposers and some are symbionts. Filaments with septae and dikaryotic, you understand that? And then we can see, people, the dikaryotic mycelium is the dominant stage of the life cycle. They produce fruiting bodies. Forgot to mention that. What is the fruiting body here? This one is the fruiting body. What do you call the fruiting body? The name Basidio carp. That is basidiocarp. Basidiocarp is the fruiting body. Basidiocarp is the fruiting body. Right? We can see that there is a fruiting body forms here and then produce exogenous basidiospores. Asexual reproductive structures are not very common. Asexual reproductive structures are not very common. Right? Asexual reproductive structures are not very common. All right. Asexual reproduction. So that is about, people, basidiomycota. And people, our paper is based on cell structure function 2. Cell structure and function 2. We talked about cell cycle recently. Right? Cell cycle we discussed recently. Now, Saturday class, people, we have little more remaining part of the animal diversity, but it's going to be like an hour work, 1 hour work, small part, and then we have to finish the tute. I have special information. You have to finish that tute. Haridha, plant animal diversity, I have given you a tute. Answer it and come, because there are a lot of questions, right? So we can start plant at least after the interval. Right? If you finish the tute and then be ready and come, people, we can finish it and then start the new lesson. Right? People, online people, I have posted the plant tute. We'll be sending it now little earlier, right? We'll take our best effort to send you the tutes earlier, right? And then people, how about we go through a little bit of cell structure details and talk about it, because we talked about the microscope, cell structure, people, cell structure long once upon a time, cell structure. Once upon a time, we talked about cells. What happened to my book? Once upon a time, we learned, people, about the cell structure lesson. If you couldn't learn cell structure lesson, you have good news. You can join our 2028 class, people. We are starting soon, 2028. Those who have missed those who have forgotten the cell structure and all, they can join with the juniors. Come and learn introduction to biology with the juniors, right? And then chemical basis. If you are interested, you can come and see our juniors, right? The juniors are coming. You all are seniors now, right? Juniors are going to come. You can learn these early lessons once again. Good, right? You can recommend. Me, to some of your juniors,
say that there is a mad teacher. Yeah, send a couple of them to me also. Cell structure function. Cell structure function. So when is the 20th class? 20th class is on a Monday. Monday morning from March 9th. We are starting on March 9th. March 9th. We'll give you a handbill. March 9th morning. 10 to 12 initially, to 10 to 1 o'clock, I think. 10 to 1. Then later it comes to Saturday afternoon. Right, people, we were discussing about this cell structure lesson. Cell structure lesson. Where did we start? Cell structure. We talked about, people, the instruments, organization of cells. Organization of cells. We learn a lot, ne? It's a long lesson. Ne? Somebody said the cell structure lesson is not confident. A new student said not confident. I think you have forgotten it long time ago. Ne? Contribution of the microscope to the expansion of the knowledge of the cell organization. We learn, people, there are two types of microscopes. Ne? The light microscopes are there, right? So is it the same class number 19 for the next class? Yeah. Now 19 is fixed for us. I have sorted out that issue. Our uncle, toilet uncle, the uncle who's not letting you to go to toilet and canteen, I sorted it out. And you're not going to have any problems hereafter, right? So talk to him nicely, okay? And that problem won't be there anymore. If anything, something happens, come and let me know, right? There was a small issue, you didn't tell me, but the afternoon people said that that the card marking uncle is not letting you to go through, right? I talked to him, right, people? So contribution of microscope to the expansion of knowledge in the cell and the cell organization. We learn two types of microscopes, people. We learn light microscope and we learn about electron microscope. Light microscope use a beam of light. It's the early type of microscope. There was a long history of the development of the light microscope. Zurcharia, Janssen, and then we talked about, people, Anton van Leeuwenhoek, and then the contemporary Robert Hooke, right? Contemporary of Anton van Leeuwenhoek was the Robert Hooke, and their microscopes in the 1650s, people. 1650s, and then gradually this improved further with time, and then people start using more and more better advanced instrumentation to study the cells. So light microscope, we learn, people, simple light microscope, dissecting microscope, student microscope. We said that they are light microscope, sometimes single lens, sometimes many lenses are there. Compound light microscope, we talked about it. It has two lenses. Actually, people, it has two lens sets. It has two lens sets. The compound light microscope. That those are the what do you call the those are the eyepiece lens and the objective lens. Eyepiece and the objective lens. Compound light microscope. Eyepiece and the objective. So we learn, people, the microscope is magnifying images. So these lens set is there, the lens set is there to magnify images. You learn, people, these lens sets, their magnification values are different. So we name them, people. We name them differently. What do you call these lenses? You call these lenses low power lens, medium power lens, high power lens, like that. Different lenses are there, right? And then here, people, we hold the glass slide. Some structures are there to hold the glass slide. And then you learn, people, there is a condenser lens beneath. The Condenser lens is there. So we learn parts of the microscope, right? Yeah, we learn. This is you name them. What do you call these people? This is the eyepiece. And then people, ocular tube, ne? This part is the ocular tube. What do you call them here, people? Objective lenses. 3 objective lenses are there. We call 10x medium power, 4x the low power, 40x the high power, like that, ne? Right? Then how do you get the total magnification of this instrument, people? Eyepiece into objective lens. Total magnification, we take, people, eyepiece into objective lens magnification. So if you are using an eyepiece of 15x and high power of 40, what is the total magnification at that moment in the instrument, people? How do you calculate that? 600, 615 into 40, right? So suppose you have an eyepiece, you can have an objective lens. Sometimes the magnification can be around 100. So 100 into 15x is kind of like the board. What we said, the maximum magnification? What we said, maximum magnification? What we said, the maximum magnification, 1000 times. 1000 times is the maximum magnification. We talked about something called as a resolution power. Resolution power is a minimum distance that should present between two objects to distinguish them as two. That's the resolution power, the minimum distance that should present between two objects to distinguish them as two. Resolution power. Resolution power is, what is the value? I can't remember resolution value of the light microscope. 0.2 kilometers. 0.2 nanometers. Oh, who's that? 2 nanometers. Oh my God. Micrometers, dear. Micro. 2 nanometers for the electron microscope. 2 nanometers for electron microscope. Not for that. Light microscope. If the light microscope can give 2 nanometers, you can see DNA using the light microscope. That's going to be awesome then. Oh, light microscope. We can see DNA also using that. DNA is 2 nanometers. So you need the electron microscope for that. 200 nanometers is correct. 200 nanometers is correct. Hope you remember the units. And then people, coarse focus control, fine focus control. We talked about the sample preparation, people. We first, how do you use this microscope? Question. How do you use the microscope? You first turn on the low power, right? Look through the microscope and adjust the mirror, right? And you place the slide, and then looking through the microscope, you move the slide and adjust it. And then people, you turn the coarse focus control, fine focus control, and then you try to sharpen the image. You want the medium power, turn the medium power on. You want the high power, turn the high power on. So that's the procedure of using a microscope. We can observe, people, different specimens using a light microscope. You can take some cheek cells. You can scrape some cells from your cheek and you can observe them, people. In the cheek cells you can observe. And then we said pond water sample, and then people, we said something, hay infusion. Hay infusion. What is hay infusion? Hay infusion, we can observe hay infusion, right? What is hay? How do you prepare hay infusion? How do you prepare hay infusion? Royo, yeah, royo. Amoeba, paramecium, euglena, we observe. What is hay infusion? Somebody remembers what hay infusion is? Can somebody remember hay infusion? Straw in water, that's correct. Straw in water, but that water is pond water. You take pond water into a jam bottle, and then you add some decaying grass into it, keep it for about 2 weeks time period, and then you get a hay infusion. right? You add a little bit of salt and then you can drink it. How about that, right? Drinking that. So you'll get diarrhea after that. So congratulations, do that. You can prepare a hay infusion, and the purpose is to let these microorganisms to grow in some nutrient containing medium, right? We have observed, look, Danisha says we have observed. School, you did that? School, nice. You did it recently. Now only you are doing those things in school. Mm, no, long time back. Good. And right, hay infusion. So those are the things that you need to know regarding 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 um, the microscope, light microscope. Resolution power is a minimum distance between 2 points that can be distinguished. Moving to stucking. Magnification is limited due to the resolution. Light from an object pass first through the objective lens and then produce magnified image. Above image then act as an object for the second lens. The total magnification, how you work it out. And then the electron microscope. Now, electron microscopes were discovered, people, long time after the discovery of the light microscope. 1930s, Ruska and Noll, two German scientists, they discovered, actually they developed a new form of microscope that uses a beam of electrons, because they realized the light itself is having a limitation. You learn, people, wavelength half is a maximum resolution power. Neither resolution, when you have the perfect lenses, when you have the super lens set, right? Finally, what determines the resolution is the wavelength half. You think of the best set of lenses that we can have for a light microscope. When you set them up, then the final determining factor of the resolution power is the wavelength of the beam, right? So the wavelength itself is having a weakness. So what do you do, people? You have to change the wavelength. When you are changing the wavelength, you change the beam. So you have to use some different beam. You can't do this job with the light, right? Therefore, scientists realize it's not going to work with light every time. So they develop a new microscope that we use, people, electrons instead of light. And we call that microscope as the electron microscope. And you have electron microscopes at school. Did you observe any specimen using electron microscope in school? We observed something in the class using electron microscope, didn't we? We did that, electron microscope. One day I brought an electron microscope to the class. Didn't I? No, you don't remember? I saw only hours. Right. Electron microscopes you can't bring into a classroom. It's a huge instrument, and then you need some special environment for the electron microscope, right? So in this developed world, so you brought through you brought it through a projector. Yeah, I showed it using the projector. And electron microscopes, two types of these microscopes are there. We say transmission electron microscopes and the scanning electron microscopes. The purpose of these electron microscopes are different. Transmission electron microscope is studying the internal structure and the What is it? Right, transmission electron microscope to observe internal structure. Scanning is basically shows you the surface details, right? So special sample preparation is there, people, for the electron microscope. Special sample preparation, 3D image can be observed. Special method is there for the sample preparation. We have to dehydrate the specimen. Very thin sections need to be taken. And then people, the heavy metals are going to be used for the staining process, right? So this is going to kill the specimen. Only black and white images can be observed. No colored, no live specimens. You can't directly focus electrons to your eye. So you can't see the image to your eye. You can't see it to your eye. You have to take people what? You have to take the micrographs. You have to use the micrographs, right? So if you try to focus electrons to your eye, it basically does not enter into your eye. Electrons can't pass through air, electrons can pass through water, electrons can pass through glass, and not even through your eye, right? So it's foolish thing to design to focus electrons to your eye, because you can't do it. Electrons are not going to come to your eye. So therefore, people, we have something different. We have a huge chamber like this, which we call as the electron beam passes through this cylinder. Electron gun, the upper part is electron gun. And then people set off lenses are here. What do you call these lenses, people? Electromagnetic lenses. The electron beam is going to pass through these electromagnetic lenses. Electron beam is going to pass through these electromagnetic lenses. They're going to pass through electromagnetic lenses. And finally, people, electrons are going to be focused on the, so the tungsten filament is there. The electrons are going to pass, and finally, people, they are going to emit on the screen here. What do you call the screen, people? Fluorescent screen. So you get black and white image there. If you want, you can watch that from a side. You can watch that from a side, but you can't focus. Inside of this instrument is a vacuum. There is a special preparation, sample preparation procedure, right? If you want to do it, you have to do it carefully. So we say, people, transmission electron microscope used to study internal structure of the cells. Beam of electron is passed through the thin, specially prepared slice of material. A very thin specimen is used, specimen stained with heavy metals, which attach more to the certain cellular structures than the other areas. Image reflects the pattern of electron passed through the specimen, displays on the screen. While electrons pass through the specimen, more electrons get displaced. That's all. Where the structures were densely stained. That's important part. If you want to see your specimen, has to be a thin one. Your specimen has to be a thin one, and once you stain it, the heavy metals are going to absorb more to the wall and more to the nucleus, less to the rest of the part, which gives the contrast. So if you want to get the contrast is this color difference, right? So therefore, people, we can see that you must use some stains. Heavy metal stains you have to use. So that is about the transmission electron microscope. Scanning electron microscope is different, where you observe the reflection. You observe the reflection, the beam which reflects from the specimen is observed. Look at this, people. This microscope has a chamber, vacuum chamber like before. And then you don't have to take thin sections of your specimen. You can put the whole specimen here. You can put the whole specimen inside, and then there is an electron gun
shoot electrons. The specimen is stained with what do you call gold? What is this dye, people? What is this dye we used to stain? What's the dye that we? No. What's the dye? Gold, eh? Gold. And then people, these electrons are going to reflect. The reflected ones are captured by the detector. Detector send this information to the computer monitor. And then people, you can see through the monitor. You can see through the monitor. So that's how it actually works. That's how it actually works. Right? The, what do you call this? This is the scanning electron microscope. What are the uses of this scanning electron microscope? People, like a document scanner, this is going to show you the surface details. Can you see internal structure using can you see the internal structure using this microscope, people? You can't see internal structure. Three-dimensional weave you can observe. That's like an extra advantage. Three-dimensional weave can be observed, right? 3D weave can be seen. That's nice. And 3D weave can be observed. Surface details can be observed. So those are the uses of the microscope. So check this, people. Used to study internal structure of the cells. In this microscope, a beam of electron is passed through a thin, specially prepared slice of material. A very thin specimen is used. Specimen stained with heavy metals which attach more to the certain cellular structures than the others. Image reflects the pattern of electron passed through the specimen, displays on a screen. While electrons pass through the specimen, more electrons may get displaced in regions where the structures are densely stained. We said that. Sorry. Scanning electron microscope. This instrument find a fine beam of electrons reflected from the surface of the specimen. Specimen is mostly coated with gold prior to observation. Here the specimen scatters many electrons, and where others are absorbed, the instrument is ideal to observe the surface details. Light microscope and the electron microscope, differences are there. What sort of differences are there? Glass lenses, powerful magnets. Image is directly detected by naked eye. You need micrographs. And then people, you can see living and non-living objects, only non-living. Actual colors can be seen in light microscope. You cannot see that. Dyes are used to stain. Heavy metals are used to stain, right? So those are the things. Those are things. I want you to go through, people. I want you to go through the part, cell structure and function. If you find it difficult or if you need any help, people, I'm ready to discuss this lesson further with you. But dear, not today. Not today, right? Some other day, right? We are having the paper discussion next Thursday. Saturday, people, as usual, our regular class, right? Okay, then thank you very much for your participation today, right? And then we'll meet you, people, right, in our regular weekend classes, right? Thank you very much and have a nice time. Salute for everyone. Bye-bye. See you.